Welcome friends, I am Dr. S. Megala, Assistant Professor, Department of Microbiology, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I am going to talk about algae. Algae. The algae are simple chlorophyll bearing plants. They are mostly unicellular microorganisms. Others are aggregations of many similar cells. The study of algae is called phycology. Algae are heterogeneous group. They vary in size, habitat and reproductive process. The size ranges from microscopic to several meters in length. Algae are abundantly present in seawater, freshwater, on rock stones, barks of the trees, on plants and animals. Algae occur where there are sufficient light, moisture and nutrients. Algae are chlorophyll bearing autotrophic organism in which the sex organs are unicellular or multicellular. The plant body of algae is called thallus. It does not show differentiation into root, stem, leaf and true tissues. The common example of algae are Chlamydomonas, Volvox, Chlorella, Spirogyra, Ulothrix, Oscillatoria, etc. They are either free-living or attached form. The free-living form called plankton. The algae are attached to rocks are called lithophytes. The algae attached to plants are called epiphytes. The algae attached to animals are called epizoophytes. The classification of algae, they are three main group of algae being green algae, red algae and brown algae. First one, green algae, habitat, freshwater, marine water and terrestrial. Photosynthetic pigment, chlorophyll A and chlorophyll B and carotenoids. Cell wall components, cellulose. Storage food, starch. Flagella, 2 flagella per cell. Asexual reproduction, reproduce vegetatively by fragmentation. Sexual reproduction, production is isogamous, ensiogamous and oogamous. Example, Chlamydomonas, Volvox and Ulothrix. Second one, red algae, habitat, freshwater and marine water. Photosynthetic pigments, chlorophyll A and chlorophyll D and carotenoids. Cell wall components, cellulose. Storage food, Floridian starch. Flagella is absent. Asexual reproduction by fragmentation or by non-motile mitospores. Sexual reproduction oogamous. Example, Gracilaria and Gelatium. Third one, brown algae, habitat, freshwater and marine water. Photosynthetic pigments, chlorophyll A and chlorophyll C and carotenoids. Cell wall components, cellulose and alginic acid. Storage food, mannitol. Flagella, 2 unequal flagella are present. Asexual reproduction by fragmentation or by motile-zoospore. Sexual reproduction. Reproduction is isogamous, ensiogamous and oogamous. Example, Ectocorpus and Laminaria. Next, isolation of algae from soil sample. Blue-green algae constitute an important group of microorganisms to fixing atmospheric nitrogen. They are photoautotrophic microorganisms which utilize solar energy to fix atmospheric nitrogen. A rice field ecosystem is a favorable environment for the growth of blue-green algae, and they fix about 25 to 30 kg of nitrogen per hectare per cropping seasons. Fixation of nitrogen takes place in specialized cells called heterocysts. The nitrogen fixing heterocytous blue-green algae commonly found in the rice field include Nostoc, Anabina, Calothrix and Toliphothrax. Materials required for the isolation of the algae: soil sample, sterile water, test tubes, conical flask and Fox liquid medium. This for nitrogen-free Fox medium composition. Next one, procedure for the isolation of the algae. About 1 gram of sample from a rice field and transferred to a test tube containing 9 ml of distilled water and mixed well to get 10 to the power of 1 dilution. 1 ml of homogenized sample from 10 to the power of 1 dilution is pipped out into a test tube containing 9 ml of dilutants using a sterile pipette to get 10 to the power of 2 dilution. The procedure is continued using sterile pipettes for each dilution up to 10 to the power of 5 dilution. This liquids of appropriate dilution 10 to the power of 2 and 10 to the power of 3 into free nitrogen-free liquid Fox medium in a conical flask. Incubate for 3 to 4 weeks in a illuminated growth chamber or in partial sunlight at 28 to 32 degrees Celsius to observe the algal growth. Purification of the algae. Take the individual colonies with the help of the inoculation needles. Vigorously shake to make a homogeneous suspension. Add 0.5 ml of the suspension on an agar to prepare seeded medium plate and incubate in light for a week. Examine the developed colonies under microscope. Transfer the algal culture to Fox agar slant for further growth. Thank you.